My name is Jatin Roper. I'm a scientist in the Yilmaz lab at the Koch Institute at MIT, and I'm a gastroenterologist at Tufts Medical Center in Boston. A major clinical problem that I see in my practice is colorectal cancer. Colorectal cancer is the third leading cause of cancer mortality, and 50,000 Americans die each year from colorectal cancer. In the lab, we are interested in studying how conditions such as aging and obesity affect the formation of colon tumors. Currently, we study patient colon cancers in mouse models. However, the available mouse models are limited because they are time-consuming to produce and often do not represent key features of the patient disease. Therefore, we use three-dimensional cultures called organoids. These organoids are a great model of intestinal function because they are faithful to the structure and the function of the intestine. However, these organoids have some limitations in that they cannot tell us how the intestinal cells interact with other cell types in the intestine, for example, inflammatory cells and bacteria that live in the intestine. We collaborated with Thomas Tomella from Tyler Jacks's lab here at MIT to combine the virtues of the organoid cultures and the mouse models by injecting these organoids directly into the inner wall of the mouse during colonoscopy. In the image, what you're seeing is a microscopic focus of a mouse colon, shown here in blue and green. We then transplanted the organoids derived from a different mouse, shown here in pink. As you can see, these transplanted organoids are successfully engrafted into the recipient colon, and other studies have shown that these organoids can continue to divide and replicate and fully function as intestinal cells in the recipient colon. In addition, we are able to transplant mouse cancer organoids. We have found that these transplanted cancer organoids rapidly grow into tumors in the recipient mice. We are very excited by this new technology because it allows us to perform experiments that previously were not possible. We are harnessing a new approach called CRISPR-Cas9 to edit genes both in organoids prior to transplantation and in the mouse colon directly. By doing this, we can understand how different genes influence the formation of colon cancers. Second, we are using the injection approach to study how normal colon tissue develops into cancer in conditions such as aging and obesity. And finally, we are using these technologies to understand which pathways in cancer cells are most important in cancer progression in order to develop novel therapies for colon cancer and hopefully one day improve the care of patients with colon cancer.